Welcome to Unit 2, Topic 8. Today we're going to be discussing tonicity and osmoregulation. To begin, let's do a quick review of osmosis, look at both of these questions, formulate a response, and then we'll go through them together. All right, let's go ahead and start with the first one. What is osmosis? Remember, osmosis is the diffusion of water down its concentration gradient, or you can think of it as the movement of water from a low solute concentration to a high solute concentration, which leads us right into question number 2. Here on the left-hand side of the U-shaped 2, we see less solutes, less of these purple circles, than on the right-hand side, so we'll see water move to the right-hand side. All right, now let's go through water balance, and let's start with tonicity. So tonicity is the ability of an extracellular solution to cause a cell to gain or lose water. And the key here is really that it's an extracellular solution. It's not the cell, it's a solution that the cell is placed in that will cause it to either gain or lose water. Now, this will depend upon the concentration of solutes that cannot pass through the membrane, and these are called the non-penetrating solutes. Now, cells can be placed in 3 types of solutions: isotonic, hypertonic, and hypotonic, and we'll go through each of those in just a minute. But first, let's talk about osmoregulation. So all cells must be able to regulate their solute concentrations and maintain water balance. But cells will do this differently. So for example, animal cells will react differently than cells with cell walls, like plant cells or fungi and some protists. So now let's look at tonicity in action, and we're going to go through each of the 3 solutions. We're going to start with isotonic solutions, and notice at the bottom, for each solution, I'm going to be using illustrative examples. So the animal cell, this is a red blood cell, and then this is a plant cell. So we'll be comparing what happens to each of these in each of the 3 solutions. So in an isotonic solution, so cells that are immersed in this type of solution will have no net movement of water. And this is because the concentration of those solutes that cannot pass through the membrane are equal inside of the cell and outside of the cell. This means that water will diffuse into the cell at the same rate that it moves out of the cell. Don't think that water does not move in isotonic solutions. It does. It just has no net movement, meaning it moves equally into and out of the cell. Now, if we look at the animal cell and the plant cell, both do well in an isotonic solution. Now let's go ahead and look at another image, but focusing on the solutes. So here you can see all 3 solutions, and we're going to look here at the center for the isotonic solution. If you count the green solutes, the blue here, that's outside the cell, and the orange is the cell. If you count outside of the cell and inside of the cell, you'll notice that they're equal. So the solutes inside and outside are equal, which would mean water is going to be moving into and out of the cell equally. So out of the cell and into. I realized I said that backwards when I drew the arrow. Sorry. Outside of the cell and into the cell equally for water. So that's an isotonic solution. Next, let's go ahead and look at a hypertonic solution. So cells immersed in a hypertonic solution lose water to their extracellular surroundings. And that's because the concentration of those solutes that can't go through the plasma membrane are higher outside of the cell. So this means water will move to the extracellular fluid. And for both animal and plant cells, the cells will shrivel, and they have the potential to die depending upon how extreme of a hypertonic solution is or how long they're in the hypertonic solution. So let's look at the animal cell first. So here we can see it's much smaller in size from the isotonic solution, which is the normal size of a red blood cell, and water is leaving the cell. Then if we look at the plant cell, we also see water leaving. And when water leaves, the key here is that this central vacuole shrinks. So if you compare this to the previous image of the central vacuole, it's gotten much smaller. So when that happens, the cell goes through a process called plasmolysis. So when this central vacuole shrinks, it's going to cause a plasma membrane to pull away from the cell wall. And we can see that here. So this is the plasma membrane. You can see it's not up against the cell wall anymore. It's pulling away. If let's say you don't water a plant for a long time, you'll notice that the plant gets quite droopy and it's not standing upright. And that's because the cells are going through plasmolysis. That central vacuole has shrunk, and then that plasma membrane pulls away from the cell wall. All right, now let's go ahead and look at an image, again, focusing on the solutes. All right, so here we go. So here is the hypertonic solution. Go ahead and look at the outside. Notice we have much more solutes here in the blue or outside of the cell than we do inside. Now remember, water goes from the low solute to the high solute in osmosis. Or you can think about it in water itself. Here we have more water because there's less solutes, so water will go from the higher concentration of water to the lower concentration of water. All right, and then lastly, we have hypotonic solutions. So cells immersed in a hypotonic solution will gain water, and that's because the concentration of non-penetrating solutes is lower outside of the cell. So the cell will gain water, and for animal cells, they'll swell and lyse, and we can see that here. Look how much larger the red blood cell is now, and that's because water is going into the red blood cell, and we can see the cellular contents being burst out right there. So that's an animal cell, so they'll swell and lyse. Plant cells, on the other hand, work optimally in a hypotonic solution, and it's all because of this central vacuole here. It can take up a lot of that water. It puts pressure on the cell wall. Remember Turgor pressure, and that's how plant cells like to be. So plant cells can survive in both hypotonic and isotonic solutions, but animal cells can only really survive in isotonic solutions. Let's go ahead and look at it again in terms of those solutes. Whoops, there we go. So here's our hypotonic solution. And in this one, we can see that the solutes are higher inside of the cell than outside of the cell. And so water will move into the cell. Again, you can think of it as going from low solute to high solute, or water moving down its concentration gradient. Okay, but that's it for tonicity. Now we're going to shift focus and talk about water potential. So what is water potential? Well, water potential is a physical property that predicts the direction water will flow. And it takes into account not only solute concentration, but also the physical pressure placed on our solution. And I'm going to give you some just things to really memorize. It's most helpful when you memorize this going through practice problems for how water will flow. So firstly, water will flow from areas of high water potential to areas of low water potential. Water will flow from low solute areas to high solute areas. And then lastly, water will flow from areas of high pressure to areas of low pressure. So again, just memorize these, have them written somewhere that you can refer back to, and just practice going through these so that you can quickly identify in problems how water will flow. Now let's go ahead and go through the formulas for water potential and solute potential. And we'll start with water potential. So here's the formula for water potential, and this symbol here is the Greek letter psi, and that represents water potential. And it can be measured in megapascals or bars, and here's a conversion for those so that you have them. I've never seen the AP exam ask you to convert it, but just in case, 1 megapascal is 10 bars. Okay, this symbol here is for solute potential, or you can also refer to it as osmotic potential. And an increase in solutes causes binding to more free water, and therefore that's going to reduce water potential. And solute potential is always going to be expressed as a negative number. And something that I need you to memorize is that pure water is 0 megapascals or 0 bars. And then lastly, pressure potential. This is the physical pressure on a solution. It can be positive or negative, just depending upon the atmospheric pressure. And again, something to memorize is that open air means 0 megapascals or 0 bars. So that's a formula for water potential, but we also sometimes need to calculate solute potential. So sometimes they won't give us this. So how do we calculate that? Well, we're going to use this formula here. This, again, is our solute potential, measured in megapascals or bars, like we said. This here, I, negative, it's an ionization constant, and it represents the number of particles formed in the solution. All right, what does that mean? Well, let's say we're dealing with something like NaCl. If we put NaCl into a solution, it can form two ions, right? It can form our sodium ions, or it can form our chlorine ions. But sugar, if we put, let's say, sugar in the solution, it doesn't form any ions. So all you're doing is trying to figure out how many particles are going to be formed. So in the case of NaCl, we form two particles. If no ions are formed, then it's one, and sugar is a good example. And the reason I use both of these is because these are the only two examples I've ever seen them use on the AP exam. So they'll either give you NaCl, which will form two, or sucrose, which is just an ionization constant of 1. Okay, moving on, C, that's our molar concentration. R is our pressure constant, and there's the numbers for those. Again, you don't need to memorize this. AP exam will give it to you. And then T is temperature in Kelvin, and to calculate Kelvin, you do 273 plus the degrees in Celsius. All right, so those are both of our formulas. Now let's go through some practice problems, and I have 5 for us to do. So let's look at the first one. So go ahead, take a minute, read this, and we'll come back together. All right, so this problem is asking which way water will diffuse if the cell is placed in a 0.3 molar NaCl solution. All right, so let's just do a little drawing. Do it down here. All right, so here's my cell right here. My cell, it says, is 1.15 molar NaCl. And then the solution that it's placed in, so here's my solution, is 0.3 molar. Okay, well, let's go back to what I asked you to memorize. How does water move in terms of solute? Well, it goes from low solute concentration to high solute concentration. Here, the cell is lower, so I would expect water to leave and go into the solution. And there we go. Water will leave the cell. It'll go into the surrounding solution. Okay, let's look at our second practice problem. Go ahead, take a minute, read this, and try to do it on your own, and then we'll come back together. All right, so this one is asking us to calculate the solute potential at 27 degrees Celsius and to find our answer in bars. So we'll use this formula. Now let's go ahead and figure out what each of these is. All right, well, firstly, they tell us that it's a sugar solution. So we know negative I is going to be 1. C, our molar concentration, well, it tells us this is the concentration here. So this is going to be our C. R is our constant. So next we need to look at our temperature. So we'll have this plus 273. So if we do all of that, there you go. That's what you should have sort of plugged into the formula. So if we plug that into our formula, you should have gotten that our solute potential is negative 7.48 bars. Okay, let's look at problem number 3. Go ahead, look at this. It's asking us to go back to practice problem number 2. So I'm going to go back to it, but now I want you to solve for water potential. Okay, so using this information, go ahead and solve for water potential now. Okay, well, water potential, that's our formula. So we know what solute potential is, and then since it tells us that our beaker is in the open air or out on the lab desk, that means open air, so we know that the pressure potential is 0. So if we plug that in, our water potential in this case is also going to be negative 7.48 bars. Okay, let's look at number 4. Go ahead, read through this, try to calculate an answer, then we'll come back together. All right, so here we're looking at solute potential again. So here's our formula. In this example, this is going to be our C, so we know the molar concentration is 0.2. NaCl is going to form 2 particles, so negative I is going to be negative 2. And then we know it's at 30 degrees Celsius, so we'll do that plus 273 to get Kelvin. So that's what you should have plugged in there into your formula. And if we plug all of that in, you should have gotten a solute potential of negative 10.07 bars. All right, so now let's go ahead and look at number 5. This is a 2-step problem. That's your hint. So go ahead, take a minute, calculate this, and then we'll come back together. Okay, so firstly, we need to figure out water potential. But in order to figure out water potential, we have to figure out solute potential first. So we're looking at the solute potential of the solution. So let's go ahead and sort of read through this. So it says, if you place this root tissue in 0.2 molar sucrose. So this is, I know, my C, my molar concentration. Sucrose means the negative I, the ionization constant, will be negative 1. And then we have the temperature as well. And we'll do that plus 273 to get it into Kelvin. So that's our first formula that we'll use. And then that's what we obtained from the problem. So if we plug all of that in, you should have gotten negative 4.87 bars. And now we can go ahead and solve for water potential. Now, since it's in an open beaker exposed to the open air, we know that the pressure potential is 0. So we'll just add the solute potential and then the pressure potential. So our water potential is also negative 4.87 bars. Okay, so now let's just take a minute and assess where we're at before we answer the last question, which is which direction will the net flow of water be? All right, so here's where we're at. This is what we were given, that the water potential of the root was negative 4.6 bars. Now we calculated that the water potential of the solution is negative 4.87 bars. So now go back to the list that I gave you of things to memorize for how water flows and how will water flow in terms of water potential. Well, water is going to flow from, high potential to low potential. So which number is higher? Well, negative can sometimes confuse people, but this number is higher. So it's going to go from here to here, or from the root out into the solution. All right, but that is it for Unit 2.